We live in an age of unprecedented scientific advancement. One of the most profound developments is genetic engineering. It allows us to manipulate the very building blocks of life. Genetic engineering, also known as genetic modification, empowers scientists to alter the DNA of organisms. This technology has the potential to revolutionize our world in countless ways. Genetic engineering is not science fiction. It is happening now. Scientists are already using it to develop new medicines, create disease-resistant crops, and even engineer animals with enhanced traits. The possibilities seem endless, but with great power comes great responsibility. As we delve deeper into the realm of genetic engineering, it's crucial to understand its implications. We must consider the ethical dilemmas it poses. We need to ensure that this powerful technology is used for the betterment of humanity and our planet. Join me on this journey as we explore the fascinating world of genetic engineering. Imagine being able to cut and paste DNA. This is the essence of recombinant DNA technology. It is one of the foundational techniques in genetic engineering. Scientists can now isolate specific genes from one organism and insert them into another. This allows them to create organisms with new and desirable traits. One of the earliest examples of recombinant DNA technology is the production of human insulin. Insulin is a hormone that regulates blood sugar levels. People with diabetes cannot produce enough insulin on their own. In the past, they relied on insulin extracted from pigs or cows. However, this animal insulin could cause allergic reactions in some individuals. In the late 1970s, scientists used recombinant DNA technology to insert the human insulin gene into bacteria. These genetically modified bacteria became tiny factories. They churned out vast quantities of human insulin. This breakthrough revolutionized diabetes treatment. Today, millions of people worldwide rely on this life-saving technology. It is a testament to the power of genetic engineering. Gene editing is like using a molecular scalpel. It allows scientists to make precise changes to an organism's DNA. One of the most powerful gene editing tools is CRISPR-Cas9. This revolutionary technology has taken the scientific world by storm. CRISPR-Cas9 acts like a pair of molecular scissors. It can cut DNA at specific locations. This allows scientists to target and modify individual genes with incredible accuracy. Before CRISPR-Cas9, gene editing was a time-consuming and expensive process. Now, it is faster, cheaper, and more precise than ever before. The potential applications of CRISPR-Cas9 are vast. Scientists are exploring its use in treating genetic diseases, developing new cancer therapies, and even engineering mosquitoes that are resistant to malaria. The precision and versatility of CRISPR-Cas9 make it one of the most exciting tools in the genetic engineering toolbox. It has the potential to transform medicine and agriculture in profound ways. Cloning is the process of creating a genetically identical copy of an organism. This groundbreaking technology captured the world's attention in 1996 with the birth of Dolly the sheep. Dolly was the first mammal to be cloned from an adult cell. Cloning has significant implications for agriculture, medicine, and conservation efforts. In agriculture, cloning can be used to produce animals with desirable traits. These traits might include increased milk production or disease resistance. In medicine, cloning could be used to create organs for transplantation. This could potentially save the lives of countless patients waiting for organ donors. However, cloning is not without its ethical concerns. The potential for abuse, such as attempts to clone humans, raises serious ethical dilemmas. Additionally, the long-term health effects of cloning are still being studied. As with all powerful technologies, careful consideration and regulation are essential. Section 5. Agricultural Advances, Engineering a Sustainable Food Future. Genetic engineering is transforming agriculture in remarkable ways. Scientists are using it to develop crops that are more resistant to pests, diseases, and harsh environmental conditions. These advancements have the potential to increase crop yields and reduce our reliance on pesticides. One notable example is the development of genetically modified crops that are resistant to herbicides. These crops allow farmers to control weeds more effectively. This reduces the need for tilling, which can help to conserve soil and water. GM crops can also be engineered to resist specific insects. This reduces the need for chemical insecticides, which can harm beneficial insects and contaminate water supplies. Genetic engineering is also being used to improve the nutritional content of crops. For example, scientists have developed a type of rice called golden rice. Golden rice is genetically modified to produce beta-carotene. Beta-carotene is a precursor to vitamin A. Vitamin A deficiency is a serious public health problem in many developing countries. Golden rice has the potential to combat this deficiency and improve the health of millions of people. Section 6. Medical Marvels, Genetic Engineering in Disease Treatment and Drug Development. Genetic engineering is revolutionizing medicine. It is opening up new frontiers in disease treatment and drug development. One of the most promising areas of research is gene therapy. Gene therapy involves replacing faulty genes with healthy ones. This has the potential to cure genetic diseases that were once considered incurable. Scientists are also using genetic engineering to develop new drugs and vaccines. For example, genetically modified bacteria are now used to produce human insulin. This has been a game changer for people with diabetes. Genetic engineering is also being used to develop new vaccines for diseases such as HIV, malaria, and cancer. Another exciting area of research is the use of genetic engineering to grow human organs in animals for transplantation. This is known as xenotransplantation. While still in its early stages, this technology has the potential to address the critical shortage of organ donors. Genetic engineering is poised to transform medicine and improve human health in countless ways. Section 7. Ethical Considerations, Navigating the Moral Landscape of Genetic Engineering. The power of genetic engineering is undeniable. However, it also raises profound ethical questions. As we venture further into the realm of manipulating life's blueprint, we must proceed with caution and careful consideration. One of the most pressing ethical concerns is the potential for unintended consequences. Altering the genetic makeup of an organism can have unforeseen and far-reaching effects on the environment and human health. For example, the release of genetically modified organisms into the wild could disrupt ecosystems and lead to the evolution of resistant pests. Another ethical concern is the potential for genetic engineering to exacerbate existing social inequalities. Access to expensive gene therapies could create a two-tiered healthcare system. This would leave the less affluent at a disadvantage. Furthermore, the ability to select specific traits in offspring raises concerns about eugenics. Eugenics is the practice of improving the human race through selective breeding. Section 8. Future Frontiers, Designer Babies, Synthetic Biology, and Human Enhancement. As we peer into the future of genetic engineering, we encounter possibilities that seem ripped from the pages of science fiction. One such possibility is the creation of designer babies. With advancements in gene editing, parents may one day be able to choose the traits of their children. This raises profound ethical questions about human autonomy, diversity, and the very definition of what it means to be human. Synthetic biology is another mind-boggling frontier. Scientists are now able to create entirely new organisms from scratch. These synthetic organisms could be engineered to perform specific tasks. Tasks might include cleaning up pollution or producing biofuels. However, the creation of artificial life also raises ethical concerns about unintended consequences and the potential for abuse. The idea of using genetic engineering to enhance human capabilities is both exhilarating and unsettling. Imagine a world where we could enhance our intelligence, strength, or lifespan through genetic modifications. While such enhancements may seem appealing, they raise fundamental questions about human nature and the meaning of life. Section 9. Conclusion: Balancing Innovation with Responsibility. Genetic engineering is a powerful tool with the potential to revolutionize our world. It offers solutions to some of humanity's most pressing challenges. These challenges include disease, hunger, and environmental degradation. However, this power comes with a profound responsibility to use it wisely and ethically. As we continue to unlock the secrets of the genome, we must proceed with caution and careful consideration. We must engage in open and honest dialogues about the ethical implications of genetic engineering. We must establish robust regulations to prevent abuse and ensure that this technology is used for the benefit of all humankind. The future is in our hands. Let us use the power of genetic engineering to create a healthier, more sustainable, and more equitable world for all.